Assalamualaikum and good morning to Doctor. We are from Group 4. Today we are going to present on replacement and knockout gene therapy. These are our group members. First is Nurul Arifah Hidayati, me, Rabita Alia binti Rezwan, Rajeswari, and Raihan Romi Shahputra. These are our subtopic for our presentation. It will cover definitions, examples, effects, pros and cons for both types of gene therapy. What is replacement gene therapy? Replacement gene therapy is a technique in replacing faulty and missing genes, which serve to keep cell growth and division with a normal and healthy copy of the gene. For example, is the gene coding for P53 or known as TP53. The P53 gene has been mapped to chromosome 17. It codes for a protein that regulates the cell cycle and hence function as a tumor suppression. Cells in multicellular organism needs to suppress the cancer. In the cell, P53 protein binds DNA, which in turn stimulates another gene to produce a protein called P21 that interact with a cell division stimulating protein CDK2. When P21 is complexed with CDK2, the cell cannot pass through to the next stage of cell division. The picture shows the mutation that occurs in the gene. The light blue color indicates P53 protein bound to the DNA in dark blue color, while the yellow color indicates the mutation in amino acid. Next, we move to the next presenter.
Okay, so the next thing is the effect of replacement gene therapy. There are three main effects of replacement gene therapy. The first one is to replace mutated genes. So by replacing the mutated genes or the defective genes, it can help to treat certain disease, because some cells that become disease are not work or maybe work incorrectly. So by replacing them, our cell will become normal. And the second thing is to fix mutated genes. These mutated genes can cause some diseases, and it could be turned off so they can no longer promote any diseases, or maybe another of our healthy genes that help to prevent that disease could be turned on so they can help to inhibit the disease. The third thing is to make disease cell more evident to the immune system. In some cases, our immune system doesn't attack the disease cell because it doesn't recognize them as an intruders. So scientists and doctors could use replacement gene therapy to train our immune system to recognize the cells that are a threat. The next thing is about the pros and cons, or the advantages and disadvantages of using replacement gene therapy. There are some advantages that we could get from gene therapy. The first one is help to replace faulty genes or adds new gene in attempt to cure disease. So by replacing abnormal genes, we can prevent the expression of the genes into certain diseases. The next is to improve body's ability to fight the disease by enhancing the function of the cell, and also it was a great prospect for treating various diseases such as cancer, cystic fibrosis, heart disease, diabetes, and AIDS. However, there are some disadvantages by utilizing gene replacement therapy. Some of these advantages is the unwanted immune system reaction, because body's immune system may see the newly introduced viruses as intruders and could attack them. This may cause inflammation, and in severe case, it can be an organ failure. And also the second thing is it can targeting the wrong cell, because viruses can affect more than one type of cell. It is possible that the altered virus, or maybe the altered virus, may infect additional cell, not just the targeted cell that containing mutated genes. It also can happen to the healthy cells. Thus, the healthy cell will be damaged and can resulting into illness such as cancer. Third thing is the infection caused by the virus. Is it possible that once introduced into our body, the viruses may recover their original ability to cause a disease? And the last thing is the possibility of causing a tumor. So if the new genes get inserted in the wrong spot in your DNA, there is a chance that the insertion might lead to tumor formation.
To knockout gene therapy. A gene knockout, which is also known as KO, is a genetic technique in which one of our organism gene is made inoperative, knockout of the organism. However, knockout can also refer to the gene that is knocked out or the organism that carry the gene is knocked out. Knockout gene or simply knockout are used to study gene function and usually by investigate the effect of gene loss. The example of knockout gene therapy will be knockout mice. There are important animal models for studying the role of gene which have been sequenced but whose function have not been determined yet. By causing a specific gene to be inactive in a mouse and observing any differences for normal behavior or physiology, researchers can determine its probable function. Mice are currently the laboratory animals species are most closely related to human, for which the knockout technique can easily be applied. They are widely used in knockout experiments, especially those investigating gene equations that's related to human physiology. Examples of research in which knockout mice have been used would include studying and modeling different kinds of cancer, obesity and heart disease, diabetes, arthritis, substance abuse, anxiety, aging, and Parkinson's disease. Millions of knockout mice are used in experiments each year.
Okay, next I will explain about the effects of knockout gene therapy. There are two effects of knockout gene therapy that I will explain. The first is reduced body weight of the sample. I use knockout gene in mice as the example for this part. From several experiments that occur, it noticed that there are several effects happen on the sample when genes knock out. Like, it will influence the body weight to be reduced. But from the scale of 100, only about 30% of the sample that got reduced on body weight. It can happen because more than 6,000 genes with null alleles that dispensable result in mice with reduced body. And for other sample, it might cause mice to be die because there are some errors that occur when gene got knocked out from their body. Apart from that, knockout gene has advantage and disadvantage. The first advantage is gene knockout help us, researchers, scientists, to understand the role of a specific gene or a region. Like if we knock out the gene, and what will happen in the body. And also, it can be screening tools in the development of drugs and can be understand the mechanism of action of a drug in the body after we attach it. Even it has a good advantage for health and pharmaceutical field and for scientists, knockout gene also has a disadvantage of the process. This method cannot be done every time because the cost is very expensive, as it requires expensive equipment and the mice cost will be high, like around $45,000 for the one experiment. And sometimes it's difficult to determine the function of gene in relation to human disease. Even though mice was chosen because it has similarity with human body, not all of the mice can be used as a reference because not all of human disease has been found in the mice body. And the last one is ethical concerns, the important thing, because even many countries has legally to use animal as research object, but recently there are some animal care organizations that animal experimentation is cruel and unnecessary, and some people think that using animal as experimental object is one of the crimes because it can cause the animal to be disabled due to failure in the experiment and will hurt the animal. Alright, that's all from us from the group 4. Thank you and have a good day.